Hey everyone, we're going to be going over some biochemistry content today. We are going between amino acids all the way down to lipids on the tree supporter packet. We're probably going to stay away from carbohydrates at least for today. I'd like to save that for later this week, so at least I can see you guys in person. This video is just a good reference guide for you guys to go back and look at because I know some of the things in here they're not overly tricky, but some small details can be mixed up, and I'd rather just have some clarification points on here because I did realize tutoring earlier this week when I was talking about the secondary structures of proteins, I could have been a little bit more clear. And so now it's a chance for me just to dive a little deeper and hopefully give you guys a better understanding of what's actually going on. So what we're going to do here is we're going to start off with amino acids. Biggest thing I want you guys to know is that just a standard amino acid by itself is going to have an amine group as well as a carboxylic acid group. Our amine group over here is going to be our basic component. Our carboxylic acid group right here, that's going to be more of our acidic component. And right in the middle, connecting all four of these little elements, including the hydrogen as well as the R group, which we'll dive a little deeper into in a second. That carbon is known as the alpha carbon or central carbon, and what it is just going to be the central point for the amine group carboxylic acid group all to attach to. So what we need to talk about here are different categories of amino acids, and these different categories are going to be based on the R group, which is right here. And what R groups do is they basically figure out what role this amino acid is going to play. So we're going to have three main categories to start off, and each of these three main categories are going to have some subgroups going along with it. Our first main category is going to be our hydrophobic nonpolar group. Hydrophobic, I know I'm probably going to sound like a broken record, but hydrophobic, the biggest thing we're looking for are C's and H's. C's and H's will carry really no charge. You've probably seen them and remember it from the lipid section to where it's just going to be a long chain of, it's basically a long hydrocarbon chain. However, here, just for the R group, we're just looking for C's and H's. And for an example, we have alanine right here with the R group just being CH3. Down here with valine, as you can see, just still C's and H's. Nothing special. But another quick topic I do want to dive into is this nonpolar section. Nonpolar means there's just no charge, and you guys might be looking at this main structure right here and like, hey, there's some charges going on. This guy's lying. But what we like to see is we both have a positive charge on the amine group and then a negative charge on the carboxylic acid group. Positive plus a negative cancel each other out, no charge overall. So for a hydrophobic nonpolar, as I said, C's and H's, nothing too special. What I would like you guys to do is, even though during exams Gutweiler will put out an answer bank at the bottom, it wouldn't be a bad idea just to memorize some of these names right here. However, when you're looking at the amino acid structures on the bottom or the R group structures they puts down there, you're going to be able to tell pretty quickly, hey, I'm only seeing C's and H's. There's no charge with it. So your first thought should be hydrophobic nonpolar. Now, within the hydrophobic nonpolar section, we're going to have a couple subcategories. We're going to have aromatic amino acids right here, and then branch chain amino acids right here. Aromatic amino acids, the biggest thing you need to know is they contain a benzene ring. And with these rings right here, each point represents an individual carbon, and there's hydrogens coming off the carbons on each one. So kind of our first thought we're thinking is, hey, it's going to be hydrophobic, nonpolar, great. However, we do need to do a little bit of memorization, even though you will be able to see the structure and the R group categories they puts out on your exam. It's not a bad bet to memorize phenylalanine and tyrosine. And also, my pronunciation is horrible, so if I butcher a couple words over the course of this video, hey, can't help it. So, aromatic amino acids, just big thing you need to know. Benzene ring, memorize phenylalanine, tyrosine, and I mean tryptophan. If you want to remember tryptophan, do it. However, I've not seen too many examples that use tryptophan. However, you never know what he's going to put on those exams, so it's never a bad bet to remember those. Last subcategory for hydrophobic nonpolar amino acids, we're looking at branch chain amino acids. I tried to come up with a trick to remember these when looking at the R groups. However, I think it's just more beneficial for you guys to understand and memorize these names right here, valine, leucine, and isoleucine. And again, being hydrophobic and nonpolar, these guys are only going to contain C's and H's. However, when you memorize valine, leucine, isoleucine, just remember that they are under the branched chain amino acid category, as well as under the larger overlying category of hydrophobic nonpolar. Same thing goes for aromatic amino acids. And what you guys might notice, I might be jumping back around, so I might lose an idea here or there, and I might just pop back up to make sure I cover everything. Continuing on, we're going to start moving into hydrophilic polar charged. Hydrophilic polar charged, or sorry, hydrophilic polar uncharged. I misread there. Basically, what we're seeing is when we think hydrophilic, I always like to think of just the H-NOF principle. It makes it very simple. Hydrogen, oxygen, fluorine, I think it is. I might be messing that up. And oxygen. But biggest thing we need to recognize here is A, polar uncharged. So our R group is not going to have a charge at all. Secondly, it's hydrophilic. So we're going to be looking for some components that could be hydrophilic. And as we can see, we have an oxygen right here in this OH structure branching off the R group. Similarly, down here, we have an oxygen right here, nitrogen right here as well. Nothing too crazy. And secondly, as well, the hydrogen bonds with these O's right here and the nitrogen, it allows them to make hydrogen bonds in water, which is a pretty cool thing. I would, again, you can probably see these in the R group structure that he will give out. However, never a bad idea to extend your knowledge and memorize serine and asparagine, or asparagus as some people like to call it. Moving on, honestly, yeah. Oh, with this group, nothing too special. So now we're moving on to hydrophilic polar charged. This is where it starts to get a little fun. So when it's talking about polar charged, we're pertaining specifically to our R group. And remember when I was talking up here about how the amine had a positive charge, carboxylic acid had a negative charge, they cancel each other out. So this charge is mainly coming from the R group. Secondly, what I want you guys to remember is when we were looking at positive, negative charges, or acidic and basic R groups, what we're kind of looking for is the extra carboxylic acid or the extra base off of the amine group, specifically in the R group. That is what we really want to understand. So if we're looking at glutamate here, it is negatively charged. And what we can also see as well is we have a carboxylic acid group right here on our R group. And what I did is drew a little bit of an extended structure right here. And this is just representing the main structure of our amino acid along with the extended R group bond. So what we can see right here is carboxylic acid. It's going to give our R group an overall negative charge. And additionally, we just want to know carboxylic acid is going to be an acidic component for that R group. Moving on now into the basic component. Basically, if you want, just pull that structure right down there. Still, we're going to have the same amino acid structure up here, and this is just pertaining again to the individual R group. And when we look right here, we have a nice little extra amine group on our R group. It's going to give a positive charge, and overall, it's going to make our R group basic. That pretty much covers it right there for acidic and basic R groups. There's a couple other categories again, or categories again. What we're going to do is, oh crap, I just lost my train of thought there. What we're going to be looking at here is additionally our sulfur-containing groups. Again, they fall under the hydrophilic polar charged category. Sulfur-containing R groups right here, and basically you guys do need to remember cysteine and methionine for our sulfur-containing R groups. Other structure, just keep that in the back of your head. However, I have not seen too many questions pertaining to amino acids with other structures. However, there will be a couple questions, or historically speaking, there have been questions where sometimes Gutweiler includes cysteine and methionine both under sulfur-containing amino acid groups in a question. So if he's asking about a sulfur-containing amino acid and he gives you both of those, pick cysteine. Cysteine is the way to go. That is the way I was taught. That's the way I've seen it a couple times, and it's usually going to be leading you guys in the right direction. Lastly, acidic amino acids here, you're just looking for that extra carboxylic acid group, or actually, no, not or. You are looking for that acidic group, and then same thing goes for basic amino acids. You're looking for that extra amine group in there. So with amino acids out of the way, we can kind of move on to the titration curve. What this is pertaining to is, for those who are not familiar with titrations, basically This little diagram, very basic, kind of demonstrating what's going on. So you're starting out with a solution, and let's say it's an acidic solution, so you're going to have a low pH. And what you're going to do is you're going to have a little titration pipette construction above the solution. And what you're going to be doing is just loading it up with a basic component and slowly dropping bits of a basic solution into the acidic solution. So if we are adding a basic element to an acidic element, over time, that pH is going to rise and create or cause the solution to become overall basic. And this titration curve is what is kind of what demonstrates what's going on. And additionally, as you guys may know, I really like buffers. This brings into the whole buffer system, everything that we've been talking about in previous units. So what I want you guys to understand is you have two pKa's present on your titration curve. And as you can probably see, these pKa's line up with these flat portions of the titration curve. What these flat portions represent is a buffer at work. And as we know, you use buffers to resist pH change. So again, referencing this example up here, this original solution right here with a low pH is kind of representing this point on the titration curve. And as we add base to it, you can see the curve slowly start to rise, and then our buffer starts to activate around this pK1, and you can see the pH rise is going to level out. This area right here is a great buffer. Great buffer, it's resisting pH change, causing a plateau in the titration curve. However, we will reach a point to where our base is starting to overcome our acid, and we're going to start shooting up right through here. This region right here and this region right here as well, they do not make good buffers, and just keep that in the back of your head. And again, continuously adding base, you're going to reach your pKa2 up here. And as you can see with pKa2, it's higher on the pH scale, which is representing we have an overall basic solution. And again, this buffer is kind of kicking in right here, helping resist pH change at that point. However, if you're continuously adding base and you already have a basic solution, I don't know how much the buffer is going to help at that point. However, we do need to know a couple things within this titration curve. You see a couple of these numbered points coming throughout this titration curve. But before we get to any of those, I want to talk about this isoelectric point right here. This isoelectric point represents a neutral charge. Zero charge whatsoever. You can also refer to it as the Zwitteron point, which Scoot Wiley likes to refer it to. Besides that, there's a couple calculations right here pertaining to our isoelectric point. If you want to find the pH of that isoelectric point, you do pK1 plus pK2 divided by 2, and that finds you this point. And again, referencing pK1 right here, pKa2, add those together, divide by 2, boom, isoelectric point. You guys might have noticed this equation right here, which is the square root of Ka1 times Ka2. If you guys remember, Ka's are basically just a precursor to pKa's, or probably didn't explain that the greatest, but if you want to find pKa from a Ka, which I believe using this pKa equation is probably going to be a lot simpler than using Ka's, you can just negative log the Ka, find your pKa's, and move on from there. However, if you want to be difficult and not do that, you can use your Ka's right here, times them together, square root, and what this is going to do is it's going to find your hydrogen concentration. And if you guys remember from the earlier math portions of this, once you find your hydrogen concentration, you just negative log the hydrogen concentration to find your overall pH, and that pH will equal your isoelectric point. Jumping back up here, as you can see, we have a couple of amino acid structures, which these are very important, and for some reason I do really enjoy talking about them, so bear with me, put me on mute, fast forward, I don't really care, I'm still going to go through with it. So what we're looking at here is we're going to look at pK1, or point number 2, and as you guys can probably tell, our isoelectric point's right here, neutral charge, and I kind of reference it to the middle of pH, so you're going to have more of a neutral pH. However, with point number 2 right here, we are in a more acidic environment, referencing this pKa kind of being lower on the pH scale. And biggest thing I want you guys to know is when we originally talked about the amino acid structure, we had our carboxylic acid group over here with a negative and our amine group over here with a positive. Overall, they canceled out each other. That is great for our isoelectric point right here for zero charge. However, if we are in a more acidic environment, our carboxylic acid is going to be pronated, we are going to have an extra H on that carboxylic acid, taking away the negative charge. So our carboxylic acid is going to be pronated, And what's going to be going on here is in an acidic environment where our amine group is going to be the only charged portion of our amino acid. Our carboxylic acid group is going to have that extra H on there, representing that the amino acid is overall a little bit more acidic in the solution. And as this progresses, we hit point two, we will see the structure, or this we will see this kind of amino acid structure more commonly in an acidic environment. And as base keeps on getting added to the solution, we're going to shoot up here to our zero charge point. And what's going to happen is as we are progressing, this hydrogen is just going to shoot off, either get picked up by some water, help form some water, but basically we just need to know that this hydrogen leaves as we go from an acidic environment to our isoelectric point. And I know I just referenced it a couple seconds ago, but our isoelectric point is neutral charge, and that is why we see our normal amino acid structure right here. We have our positively charged amine group, negatively charged carboxylic acid group. Those charges cancel out, boom, done, zero charge, isoelectric point. Nothing too special. But as we keep on adding base to this solution represented by the OH down here, we're going to become more basic overall, and another change is going to happen. So you can see down here in our acidic area, we have a positive charge on the amine group, but no charge on the carboxylic acid group. We shoot up to the isoelectric point, we lose that hydrogen, we get that negative charge back on our carboxylic acid group, we still have that positive charge on our amine group, and then as we add more base, this one of the hydrogens off this nitrogen right here is going to shoot off once again. So, what that's going to result in is a neutral amine group, but we're still going to have that negative charge on our carboxylic acid group. So I went a little bit in depth, probably got a little too wordy with it, but basically what you need to understand, going from acidic solution, in acidic solution, we are going to find that our amino acid has an overall positive charge due to this hydrogen on the carboxylic acid. As we progress to our isoelectric point, that hydrogen right there on the carboxylic acid is going to shoot off, giving us a negative charge on our carboxylic acid group right here. And then we still have that positive charge on our amine group. And then as we progress into an even more basic solution, the hydrogen on the amine group is going to shoot off, losing that positive charge for the amine group right here. And we're just going to be left with a negative charge due to the carboxylic acid group. What I want you guys to remember is just the phase changes that happen as you progress through different acidities in a solution and how the amino acid structures change as you go. Cool. Okay. We're going to keep on going. Nothing too crazy in here. This is basically just describing what I just did. So if you don't think I'm cutting it, read it for yourself. So now we're going to move into the fun stuff. Proteins. Proteins are kind of cool. They're hold a lot of functions in our body. Really, there's a lot of conformation changes that goes along with them, pertaining to like primary, secondary, tertiary, quaternary structures. We're going to get into that in a second. However, there's a main bond that links amino acids together. Again, this is our standalone amino acid. So if we want to add another amino acid onto that to make a polypeptide chain, the biggest thing we're going to need is a peptide linkage. This peptide linkage is going to occur between the carbon and nitrogen. So the carbon that I am pertaining to is not the central carbon right here. It is the carbon of the carboxylic acid. And actually, we might just hold on to that thought. I'm going to touch it. I'm going to touch back on that in a second because it's going to make it a lot clearer with this diagram that I drew earlier. But as we're linking amino acids, every amino acid we add on is kind of going to have a different name. So if we have 2 amino acids linked together, we're going to have a dipeptide. If we have 3 amino acids linked together, we're going to have a tripeptide, so on and so forth. And crap, I did kind of screw myself over here. I do have to talk about peptide linkages right now. So basically, if we're adding another amino acid on, what's going to happen is this oxygen is going to leave right here, opening up this bonding site for this carbon. And the amine group of another amino acid is going to make a linkage with that carbon right there. I'll draw it out real quick. So let's say we got NH, central carbon, hydrogen, R group. Basically what's going to happen, oxygen is going to leave. Now this carbon is going to make a bond with that nitrogen right there, and that is what we're going to classify as a peptide bond, and that is how amino acids get linked together. Shooting down Pretty simple concept I'm just going to hit real quick. If we are linking amino acids together, if we have three amino acids linked together, we are only going to have two peptide bonds holding that together because we have our central amino acid, peptide bond right here, linking another amino acid, peptide bond right here, linking another amino acid. Pretty simple concept, but never hurts to go over it. So now we're going to start getting into all these different structures of proteins and amino acids. I drew a quick diagram just to kind of walk us through primary structures and secondary structures. So with our primary structure, it's just going to be a chain of amino acids linked together by peptide bonds. As you can see right here, probably from our previous diagram as well, amino acid, amino acid, amino acid, amino acid, da da da. And each one of these amino acids is going to be linked by a peptide bond. And if you guys, I would do a quiz right now, but I am talking to myself currently. So basically, the peptide bond is between the carbon of the carboxylic acid and the amine group of the next amino acid. And overall, the main function that what happens is when these separate amino acids are binding, what's going on is that oxygen off of that carboxylic acid and two nitrogens off of the opposite amine group are going to combine to form H2O, and then as a result, a peptide bond forms right here between our carbon and nitrogen. And I hope you guys can, if you guys get a little bit confused, please just reference and kind of look at what's going on. H2O is leaving, which allows us for this peptide bond to form right here. And that peptide bond is a covalent bond. And that is the biggest bond I want you guys to know for primary structures. Is the only bond that's heavily prevalent in the structure is a covalent bond, and that is found with our peptide bond right here, linking all of our amino acids. So this reaction right here is taking place numerous times every time you link on an amino acid to create a polypeptide chain. And once a polypeptide chain gets to a specific point, we're going to move into our next structure, which is our secondary structure. I tried my best with some of these drawings, but don't make fun of me, please. I'm very sensitive. Secondary structure, biggest thing I want you guys to know is the main bonds that we're going to be seeing are hydrogen bonds. And I'll dive into that in a second, but there's another thing that we need to talk about called amide planes. And amide planes, what they basically do is they are the points of rotation for these secondary structures or these polypeptide chains as more bonds start forming. So our amide planes, what's going to happen is, see this red carbon right there? That is our central carbon from our original amino acid structure. Our central carbon, or alpha carbon as Guweiler calls it, is going to be the start and end of two amide planes. So as you can see right here, if I could draw another little carbon right here, this would be the start of an amide plane. The first bond that's going to form is the central carbon up to the oxygen of the same carboxylic acid group. I'll draw that right there. So this is its own amino acid right here. This is just a singular amino acid. This is that peptide bond we were talking about, which allows it to bind this next amino acid. What I specifically want to talk about is the central carbon right here will shoot a bond up to the oxygen of the same carboxylic acid group on the same amino acid structure, but it is also going to shoot a bond down to the next amine group on the next amino acid. And it might take you a little bit to see, but once you kind of start getting the pattern, it'll make it a little bit easier. So carbon to oxygen right here on the same carboxylic acid group, carbon to hydrogen down here on the next amine group, and then this carbon as well is shooting bonds to the same amine group, and then this carbon is also shooting bonds to the previous carboxylic acid group. And what these, as you can see, all of these bonds are starting to converge on the central carbon right here. Basically what that allows for is rotation to occur around these central carbons. And what this rotation will account for is the formation of a alpha helix. So in your secondary structure, you can have an alpha helix or a beta plate of sheet. And this is where hydrogen bonds start coming into play. So once these amide planes start to rotate a little bit, our overall polypeptide chain is going to start to rotate and twist in on itself. However, it needs some bonds to hold that structure. So what's going to happen is hydrogen bonds are going to start to form between carboxylic acid groups and amine groups. I think it was every fourth amino acid, I'm pretty sure. I could be wrong, so I will make sure to go back and after this video to double check and make sure the information I'm giving you guys is correct. However, I don't think that bond structure is overly important, or the number of amino acids between bonds is important. However, the biggest thing I want you guys to know is hydrogen bonds are the only bonds forming in the secondary structure. And what these hydrogen bonds allow for is as this alpha helix starts to contort on itself, these hydrogen bonds are helping keep that alpha helix structure. Beta plated sheets are a little bit different because it's more linear, and again, these hydrogen bonds are still occurring between carboxylic acid groups as well as amine groups on different amino acids, and this is just helping stabilize the structure. However, the drawings for this portion, I just want to make sure I could elaborate better on the primary and secondary structures, but when it comes to tertiary and quaternary structures, we're just going to dive right into it back on the Teresa Porter packet. If you guys have any questions, please feel free to reach out. I'm always happy to help. So jumping back to the Teresa Porter packet, again, we just covered primary structures, which again, only covering covalent bonds. Then we moved into our secondary structure, which only contains hydrogen bonds. But now, moving into our tertiary structures, things get a little bit more complicated because now the bonds that are forming are between the R groups of different amino acids. And there's 4 types of bonds that are going to be occurring within tertiary structures. You're going to have covalent bonds, and what these are disulfide bridges between sulfhydryl groups. Sulfhydryl groups is, if you guys can remember, are the sulfur-containing R groups for amino acids. And if you guys remember, we had cysteine and methionine, one of the two. However, actually not however, these bonds are or disulfide bridges, covalent bonds, are just forming between these sulfhydryl groups. So anything that contains a sulfur, you're going to find a disulfide bridge, and this disulfide bridge is going to be a covalent bond. And you can see that right here in this little disulfide drawing. I wish I could draw like that. However, I cannot, so I'm not even going to attempt to. I'm just going to use what's already on here. So covalent bonds, they're disulfide bridges between sulfhydryl groups of different R groups that contain sulfur. Moving on to our next bonds, we just have, again, standard hydrogen bonds. Just remember the H-NOF principle. Doesn't get more complicated than that. We're also going to have salt bridges forming. These salt bridges are only between ionic R groups. So ionic R groups, from what you guys remember, is either we're going to have a positive charge or a negative charge on these ionic groups, and basically positive gets attracted to a negative, salt bridge forms, boom, that's another bond in our tertiary structure right there. And then our last structure, or last bond, is just going to be hydrophobic interactions between amino acids. Nothing too crazy. However, if you guys remember, you guys see the word hydrophobic. I hope whenever you see hydrophobic, you get fight or flight and instinctively start thinking just about C's and H's. You're not really going to see any hydrophobic interactions between any R groups that contain O's, N's, F's, and H's. Yeah, that pretty much covers the 4 bonds that you need to know for the tertiary structures. And just a little mnemonic that we remembered is just can't help some homies. Pretty simple. However, the more practice questions you guys do, the better of an understanding you guys are going to have for the questions that are going to be asked. And I'm hoping you guys can apply this information a lot better than previously as I explained it. Then we're going to move on to our last structure for a protein called the quaternary structure. We're finding intermolecular bonds here because we're having numerous proteins come together and bind together. And the biggest example I can give when you see this is hemoglobin. Hemoglobin is it's one of the transports within your body, within your blood, that allows you to just transport oxygen around your body. Nothing too crazy when it comes to quaternary structures, honestly. I'm not seeing too many questions that pertain to quaternary structures besides the denaturation, which we were about to get into. But besides that, with quaternary structures, don't need to know anything too specific. When it comes to denaturation, just a little pretense. I don't know how many of you guys had a biology background, but this might help for denaturation. So as I said before, your polypeptide chain right here, which is our primary structure, is going to progress into the secondary structure. When it progresses into the tertiary structure, or the third structure down here, it kind of becomes an active protein. It can carry on some functions. Depending on the order of the amino acids, it has different functions depending on the different orders, but it just becomes active. And furthermore, quaternary structure is another active form of a protein. Now, denaturation it's basically unraveling the structure. So instead of going primary, secondary, tertiary, quaternary, we're going from quaternary to tertiary to secondary. However, denaturation can only affect the quaternary and tertiary structures of these amino acids. So once a protein gets unraveled back to a secondary structure, denaturation can no longer occur because the function is already disbanded. It can no longer function, and that's why people talk about how high fevers are dangerous, like 105 fevers, because significant increases in temps can denature proteins, and it can affect a lot of functions within your body. So biggest point I want you guys to know, denaturation affects quaternary and tertiary structures, and there is a nice little fun example right here that's, just kind of fun to talk about. Basically just the creation of cheese. It comes from a denatured milk protein, and if you want cheese, you just lower the pH of milk. Those proteins are going to denature, and as a result, they're going to form cheese. Keep that little fun fact in the back of your head because it is going to become important, and I hope you guys see it because that will be a good question for you guys to get a quick point on. Myoglobin, oxygen-containing polypeptide found in skeletal muscle. Besides that, nothing too crazy. So now we're getting into globular versus fibrous proteins. With globular proteins, you just need to know that it involves primary, secondary, tertiary, and quaternary structures. And the main role of globular proteins is it functions as transporters within our body. It can also function as enzymes. But again, you guys are seeing hemoglobin right here. And if you guys remember back just a section or two ago, hemoglobin is involved with our quaternary structure. A lot of this stuff kind of comes and goes and interacts with it. So you might see a couple similar concepts. So if you guys can continuously try to look out for these connections and make your own connections with them, it'll help you expand your knowledge a lot more. So globular proteins, nothing too crazy. We're going to move into fibrous proteins. Fibrous proteins right here only involve our primary and secondary structures. They're water insoluble and soluble. And biggest thing I want you guys to know is they are used for tendons, ligaments, skin, and collagen. Honestly, I was just at Costco today and I saw a lady selling, I don't even know, it might have been juice or bottled milk that contained collagen. And she was yelling at me to increase the quality of my skin by drinking this collagen. And I said, no way, Jose, I'm keeping it all natural. Sorry, that was a really crappy joke right there. But collagen right here is going to be our biggest component that we're going to talk about next in collagen synthesis. Collagen, again, as we just saw up here, is a fibrous protein. It's going to be a combination of both glycine and proline. Glycine, right out of the gate, it's pretty much ready to bind with proline right off the bat to form some collagen. However, or collagen, something like that. However, proline needs to go through a series of reactions to be ready to bind with glycine. And so we're going to talk about proline hydroxylation mechanism. And I know for you guys who are at tutoring, I talked about scurvy and a couple examples like that. And scurvy came about with sailors because they did not get enough vitamin C in their system, so they cannot repair their wounds because their collagen was weak. And the reason I'm telling you that is because vitamin C is a significant component for the proline production and the overall production of collagen. So we're going to start off here with vitamin C. He might refer to it as ascorbate or ascorbic acid. But what we need to know is vitamin C, ascorbate, and all this are reducing agents in this whole reaction. So what's going to go on is you have iron 3 plus right here. Iron 3 plus is going to swoop down and interact with vitamin C, ascorbic acid, or ascorbate. And what's going to go on is vitamin C is going to reduce Fe3 plus, and as a result, after being reduced, it is going to turn into Fe2 plus, and vitamin C and ascorbate and ascorbic acid, they are going to become oxidized and whatever. But vitamin C is just very important for the reduction of Fe3 plus. So after the reduction of Fe3 plus to Fe2 plus, Fe2 plus is going to shoot up here and go through another redox reaction with proline right here. What's going to go on is Fe2 plus is going to shoot off an oxygen to this proline to reduce the proline to get it ready for its functional structure to bind with glycine to eventually form collagen. But what you guys just need to know, Fe2 plus reduces proline, and then as you guys can see, Fe2 plus right here gets oxidized, goes back to Fe3 plus, boom, starting the cycle all over again. Besides that, though, any questions pertaining to this cycle right here, please reach out again. I'm just kind of flying through here, and I can't get a grasp on what you guys are picking up or putting down. But what's nice about this video is you guys can rewind it as much as you need. And also too, it goes through some of the mechanisms down here. Furthermore, when we're talking about cross-linking found in mature collagen, the cross-link factor could just be lysine. Just keep that in the back of your head because there very well could be a question on that, just as like a random fun fact question. However, I do not know that for sure. Now we are going to get into the Bohr effect, and I know you're probably sick of me rambling on, but we're just going to cover the Bohr effect right here. We're going to shoot through lipids and cholesterol and amniotic fluid, and we're just going to call it a day right there because I am starting to lose my voice a little bit, I think, or I'm just thirsty. That's what I really think it is. Oh, here we go. All right, we got some energy back in us. So when we're talking about the Bohr effect, the main definition of the Bohr effect is basically when the acidity increases within your body, which as a result, if you have an increase in acidity, it's going to decrease the pH. Overall, it's going to decrease the capability of binding oxygen. So that affects the overall transport of oxygen to your skeletal muscles and all that. And we're going to talk a little bit more about that. Like there's a ton of functions right in here. However, I have never really looked at any of this stuff. What I always do is flash down to the Bohr effect system right here. And what I'm going to do is I'm going to walk you guys through this whole system right here. And then we're also going to talk about these little scenarios in the lung capillaries and peripheral capillaries right there. So if you guys have already learned about this, feel free to fast forward. Or if you just want to get a little bit better, stay with me. I promise I will try and throw some humor in there. But I'm not too funny, so we're just gonna keep on rolling here. So what we're gonna start off with is we're gonna breathe in air. We're gonna start breathing in oxygen. And before I even get into that, I always forget to reference this key right here. HB stands for hemoglobin. HHB stands for hydrogenated hemoglobin. HbO2 stands for oxygenated hemoglobin, and HHbO2 is hydrogenated oxygenated hemoglobin. And what I like to reference here is this whole progression of reactions that we're about to talk about is basically like a cab system. Our hydrogenated hemoglobin right here is basically going to be our hemoglobin is basically the cab, and if you guys remember, hemoglobin falls under our quaternary structure as well as globular proteins. So our hemoglobin is going to be our main cab for transporting O2 throughout our body. So as we breathe in O2, O2 is going to bind right here with this hydrogenated hemoglobin. We're going to form into hydrogenated oxygenated hemoglobin, but we're also going to see the appearance of our bicarbonate ion, or HCO3 minus. You guys probably recognize that from the intracellular buffer system. But honestly, as we progress, what I want you guys to remember is HCO3 is our main transport for getting rid of CO2. You will see how that all comes full circle in a second. But another quick concept I want to talk on as well is, as you can see, our bicarbonate ion right here has a negative charge. And you can see right here we have a chloride ion that has a negative charge. Well, anytime this bicarbonate ion enters our lungs, we're going to have this thing occur called a chloride shift. And this chloride shift, basically what it's doing is chloride is leaving our lungs to balance out the ions going on within our lungs, so we don't have an overpopulation of negative charges. So continuing on, as I said, we have our hydrogenated oxygenated hemoglobin right here. We have our bicarbonate ion right here. As we're progressing into the next lung, we still have our hydrogenated oxygenated hemoglobin, and we still have our bicarbonate ion. What's going to happen is our bicarbonate ion right here is going to split, and we're also going to lose a hydrogen off of our hydrogenated hemoglobin right here. So as this reaction progresses, we are going to form CO2, H2O, and then we are only going to have oxygenated hemoglobin right here. So this is what allows us for our oxygen to start going into our arterial blood and transport to our skeletal muscles. And as you guys can probably tell, when that CO2 gets released from that HCO3, we're just going to breathe it out. So nothing too complicated right there, and best advice I can give is just drawing out all these reactions and kind of coming up with your own little tidbits, because I'm just kind of flying through this stuff right now, and any further clarifications we can touch on in future meetings. So HbO2, what's going to happen is HbO2, our oxygenated hemoglobin, is going to travel down through our arterial blood, and arterial blood is oxygenated, and we're going to pop down into our muscles down here. And so what's going to happen is once this oxygenated hemoglobin gets into our muscles or gets near our muscles, it's going to pick up another hydrogen right here. It's going to turn back into hydrogenated, oxygenated hemoglobin. And then when the O2 is finally getting released to our muscle cells, we're going to split into O2 and hydrogenated hemoglobin right here. Our O2 is going to shoot off into our muscles for cellular respiration. However, you guys have probably already seen this hydrogenated hemoglobin right here. This hydrogenated hemoglobin is going to shoot up through our venous blood system. And remember, hydrogenated hemoglobin and venous blood. And it's going to shoot up to the cycle right up here again, where it starts the whole process over, where it picks up the O2, and we go through all these series of reactions again to get back to our form, to get oxygen all the way back down to our muscles. So after we go through some oxidative reactions in our muscles, our byproduct is going to be CO2. So CO2 is going to be popping back out into our blood system, or I don't even know, honestly, I don't even know if our blood system for sure. However, CO2 is going to pop out, and you're going to see some H2O right there. They're going to combine to make H2CO3, and then we're going to lose one hydrogen as we progress to form hydrogen plus our bicarbonate ion. And this hydrogen is just going to shoot back up here for this oxygenated hemoglobin to hydrogenate it and to turn it back into hydrogenated, oxygenated hemoglobin. However, we see HCO3 right here, and HCO3 is going to leave through our intracellular buffer system. But when this bicarbonate ion leaves, as we see, we're going to have a chloride shift back into the muscle instead of leaving the lungs in the previous example. And this HCO3 or bicarbonate ion is going to travel all the way back up through our intracellular buffer system, pop out in this beginning stage again, and as we talked about earlier, it's going to progress through basically for the release of CO2. So that's a pretty basic overview of how the Bohr effect occurs, but there's a couple other things we got to know. So we have this little equation right here, H plus HbO2 goes back and forth or is in equilibrium with HHB plus O2. And this is basically a cab example, and my roommate liked to use the bar fight example. And basically, let's say, specifically for the lung capillaries, let's say you're hydrogen and your buddies are all at the bar, all of your hydrogen buddies are at the bar, and they're like, hey, get down here. And you're like, sweet. So you hop in a cab and you're driving down and you get close to the bar and you look outside of the cab window and you see a ton of oxygens beating up on one of your hydrogen buddies out there. Well, if you have an overwhelming presence of oxygen outside of the cab beating up on your hydrogen buddies, you're going to want to get out of that cab to equal out the playing field and go help your hydrogen buddy. So that example kind of pertains to right here. In the lung capillaries with our shift, as we can see, if we have an increase in O2 pressure or pertaining to our oxygen being in a heavier presence, we're going to have a left shift and we're going to have hydrogen disassociating from the hemo or oxygenated hemoglobin right here. And that example kind of occurs honestly, scratch that. I just had a brain fart and was trying to make a connection where I didn't know I could make one. Basically, in the lung capillaries, if you have an increase in O2 pressure here, you're going to have more dissociation of hydrogens leaving, and you're going to get a higher concentration of oxygen or hydrogen. In the peripheral capillaries, it's going to be a little bit different. We're going to have an increase of H pluses or CO2s or just an acidic component. What that's going to do is in our peripheral capillaries, it's going to decrease the pH of our blood or that area. And because if we have an increasing acidic content, we're going to have a decrease in the pH level. And as a result, if we have a decrease in the pH and an overwhelming presence here of hydrogens, we're going to have a right shift in the equation and we're going to start releasing more oxygen into our blood. Very oversimplified view. However, just looking here and being able to make some connections with this equation right here, and if we have an increase in O2 pressure, we're going to have more, this equation is going to shift more to the left, and we're going to have more release of hydrogen over here. If you can make those connections, it would be perfect. However, I'm always here for further clarification as well. So that is the Bohr effect, and the more practice problems you guys do, the better of an understanding on the type of questions we'll be seeing and the main concepts that you guys need to know and understand. Okay, so we got through that. We're going to move on to lipids here. So lipids are made out of fatty acids, or yeah, they're made out of fatty acids. And the example I was going to make right there is similarly to how amino acids combine to form proteins. It's basically the same function going on with fatty acids. Fatty acids are going to combine to form lipids, and biggest thing we need to know about lipids is they're amphipathic. So amphipathic, if you guys remember, it has a hydrophobic aspect to it as well as a hydrophilic aspect to it. So our hydrophobic aspect is going to be these long hydrocarbon tails on the fatty acid structures. And furthermore, I'm sure as we all know by now, if you see hydrophobic, your first thought is going to be C's and H's. So long hydrocarbon tail right here could be hydrophobic, but we are also going to have a carboxyl group over here on the head, which is going to be our hydrophilic end. And we can always tell that because we see some oxygens and all that right there. So right here is our head group, which will be hydrophilic. We have our tail group right here, which will be hydrophobic. If there's any questions on that, rewind or pause the video or look it up on YouTube like I did for some of the stuff to go more in depth with. I'm posting this on YouTube, so I guess, yeah. Um, well, just moving on with that. We're going to have two kinds of fatty acids. We're going to have saturated fatty acids and we're going to have unsaturated fatty acids. Saturated fatty acids is when our long hydrocarbon tails here are all of their bonding sites are filled up with hydrogens or bonds to carbon. There are no double bonds forming, and they're all the carbons are all filled up with the amount of hydrogens that they need to fill up all of their bonding sites. And as a result, that is why we call it a saturated fatty acid. Now, with unsaturated fatty acids, it gets a little bit different. We are going to have some double bonds forming in here, and as a result, we're going to lose some hydrogens. So we're going to have a little bit less of a hydrogen count in the hydrocarbon tail, but we're also going to have double bonds contorting for that. And I did just skip over a big concept here for saturated fatty acids, so we're just going to bounce back up to here and talk about it again. Saturated fatty acids, since they are saturated, it allows them to packedly together, and since they're so tightly packed and they have decent bond energy, they're going to have a higher melting point due to a higher energy. Furthermore, this is going to allow them to be solid at room temperature. So you can see that in your butters. When you cook bacon and let the grease sit for a little bit, you see that more of a solid form of the grease start to form. That is what you call more saturated fatty acids. Now, unsaturated fatty acids, since they have these double bonds occurring in here, and the fatty acid really can't rotate that well, you're going to have a lot more gaps in between these unsaturated fatty acids as they start to compile. And since they are not as solid compared to saturated fatty acids, they're going to have less bond energy. And furthermore, they're going to be liquid at room temperature. Um, and that is a big component, being able to differentiate what's going to be solid at room temperature, what's going to be liquid at room temperature. Now, another thing that we need to understand is how to count how to count fatty acids. What we're going to be looking at here is we are going to have a couple things that we need to know. So in most of the problems you will see, you are going to be given this numerical structure right here. This first number right here, this 13, is representing the number of carbons in the hydrocarbon tail right here. Do not involve this polar head group or this carboxyl group right here. All these numbers and everything are referencing the tails or the hydrophobic portions of the fatty acids. So this first number we see right here will be the overall total number of carbons in the fatty acid tail right here. And this second number will represent the number of double bonds present in the fatty acid. Now, if we see a 0, we know that there are no double bonds occurring, and that will be a saturated fatty acid, but anything else than 0, that means there is a double bond, and that means we are going to be looking at unsaturated fatty acids. Now, there's two ways to count these hydrocarbon tails right here, and you will see two different signs. You will see a delta sign and you will see an omega sign, or sorry, you will see one or blah blah blah. You'll see one or the other in the examples or problems that you will be looking at. When you see this delta sign, these delta signs means that you are counting from the carboxyl group down towards the end of the tail. And these numbers right here are representing, crap, did I preference what, hold on. I can't remember if I did it, so if I am repeating myself, I apologize. But these numbers right here, if you see a delta or omega, these are representing what carbons the double bonds start on. So as we can see right here, with delta 3 and delta 7, we can see with deltas, as I just stated, that we are counting towards the end of the hydrocarbon tail. We can see that carbon 1, carbon 2, carbon 3 starts the first double bond right there. And if we continue counting, 4, 5, 6, 7, carbon number 7 holds the beginning of another double bond. Now, let's say we wanted to count this way, we would see the omega sign because the omega sign starts at the end of the tail and works towards the carboxyl group or the polar head group. So same concept applies, just different directional counting cues. These still represent the carbons that the double bonds go on, and both of them are kind of saying the same thing, just from different directions. So if we were to count with this omega sign right here, we would go 1, 2, 3, 4, 5, 6. This 6 carbon from the tail end going this way holds the beginning of our double bond. And if we continue counting, we'll see carbon number 10 right there. Besides that, that should guys get you through counting carbons, just being able to see these cues, recognizing what's saturated, what's unsaturated, as well as recognizing which way to count for delta and which way to count for omega. Moving on, in our unsaturated fatty acids, we are also going to have cis conformations and trans conformations around our double bond structures. Cis conformation is when two components are both on the same side of the double bond right here. So CH3, CH3, they're both on the same side of the double bond. What this is going to do is it is going to put a kink in the hydrocarbon chain, and it's naturally formed in unsaturated fatty acids. Now, let's say we wanted to get these fatty acids to stack a little bit better. We would add heat to the cis conformation fatty acids, and as a result, we're going to flip our functional groups on the opposite sides of the double bond, and it is going to kind of help it pack a little bit better. However, it's still not going to pack as well as saturated fatty acids. And for the most part, that's all we need to know for that right there. So just being able to recognize that cis conformations are naturally found or naturally formed in unsaturated fatty acids, and if you want to change the conformation, you add heat, and what it does is it just flips these groups on opposite sides. Read through this if you would like. We did talk about this right here about counting and naming fatty acids. These essential fatty acids right here are going to be strict memorization, so memorize it. Now we are going to be moving into triglycerols and diglycerols. So this is when we're going to start getting compilations of fatty acids. With triglycerols, as you guys can probably imagine, we are going to have 3 fatty acids in a chain, or not in a chain, but we're going to have 3 fatty acids bound to a glycerol backbone, which the glycerol backbone is represented right here. And if we look at this triglycerol structure right here, again, right here, boom, our glycerol backbone right there. And then we have a fatty acid, and another fatty acid right here. 3 fatty acids form a triglycerol. And another thing that we need to know is that ester linkages are occurring within triglycerols. Now, this ester linkage that is kind of helping hold these fatty acids with the glycerol backbone can be broken via hydrolysis, which is just adding water to it and breaking up those fatty acids away from the glycerol backbone. But one of the major things that we have to know here is if a triglycerol does get broken up, we are going to find that we are going to get 3 fatty acids and a glycerol backbone as a result of the breakage. Secondly, you guys probably know what adipose tissue is, which is fat cells. Triglycerols are found most commonly in fat cells. So since we covered that, we're going to move down to diglycerols. Diglycerols are a little bit different. As you guys can tell, with the triglycerols, we had 3 fatty acids. Now with the diglycerols, we only have 2 fatty acids. And they're all still bound to a glycerol backbone right here. However, we have a couple subcategories of diglycerols. We have phospholipids, which you guys can probably remember. These are in our plasma membrane. And phospholipids contain 2 fatty acids bound to a glycerol backbone, but it also has a phosphate group bound to the other binding site on that glycerol backbone. So that's what helps create phospholipids. We have glycolipids, which I believe is the same concept, just like a glycerol or sugar added to that binding site instead of a phosphate. Um, Diglycerols also help with micelle formation. If you guys remember micelles, we are having a hydrophobic core, hydrophilic outside, or hydrocarbon tails are on the inside with the hydrophobic aspect, and then our polar head groups are outside, which are going to be hydrophilic. And if you guys remember, it's amphipathic agents forming together to form a lower energy form, and it helps helps transport non-soluble items out of specific areas. Um, Besides that, we're gonna have liposomes. I have not seen too much stuff talked about liposomes, but sphingolipids, we do need to talk a tiny bit about sphingolipids. Specifically, they contain nitrogen, and they are synthesized from fatty acids and the amino acid serine. Biggest thing I want you guys to know is if a sphenolipid gets broken up, you're going to have a sphericene, a fatty acid, and a variable polar head as a product. So, biggest concept I want you guys to know right there. Besides that, we really don't need to dwell too much on sphingolipids. But all of these things I just talked about in this category, just understand that they are and they go with dice or diglycerols. They go with diglycerols. And then triglycerols up here is just more fat tissue. But just make sure you are able to understand if he throws out like, oh, talking about a phospholipid, I want you guys to make that connection that is a diglycerol. Moving on now, we're moving into cholesterol. Cholesterol is a biosynthetic agent. It is also naturally occurring in your body. Right here is a major concept that I want you guys to understand is cholesterol is used to form vitamin D, bile salts, steroid hormones. Steroid hormones will include corticosteroids as well as sex hormones. And corticosteroids and sex hormones are both nonpolar, but just understand that these, I don't know, four products, because steroid hormones, I guess it includes a little extra. Just understand that these are all made from cholesterol. Secondly, cholesterol is synthesized in all cells, but is mostly made in the liver. It's not an energy source. It is an amphipathic agent. So, what I always like to know is it can go through your plasma membrane, both be in the hydrophobic as well as hydrophilic ends. And just a fun fact, cholesterol helps maintain your membrane fluidity within your cells. Um, Furthermore, stored cholesterol can be stored as a cholesterol ester. However, this mainly pertains to HDL. HDL is our good cholesterol. LDL is kind of our bad cholesterol. Now, I mean, you can have a little bit of it in your body. However, if you were just hammering the junk food or hammering and not eating your Honey Nut Cheerios, when LDL starts getting high, kind of gets a little dangerous. Not the greatest thing for you, but HDL takes cholesterol out of the blood and it is made into a cholesterol ester. Remember cholesterol ester because I'm sure you'll see a practice question on that. I did kind of fly through this, so please, any more questions, please just shoot me a text or something. Last topic we're going to talk about here, and I'm sure you guys are getting tired of hearing me talk for, I don't even know how long I've been talking for. Feels like an hour or two. But what you guys don't know is I've been watching geese and ducks out my window this entire time, and they're pretty sick. I mean, I moved here this past summer and watched some geese have some babies and watch their ducklings or gooselings or whatever you call them grow up. And I hope these are the kids and they're going to start the whole cycle all over again. And I am going to be a happy camper because nature is beautiful. And yeah. um, all right. So last thing we're going to talk about here, amniotic fluid. Amniotic fluid is present during pregnancy. Biggest thing we're going to have or need to understand is phosphatidylcholine. Phosphatidylcholine is a surfactant. So basically, what a surfactant does is it lowers the surface tension of water. What's going on inside your body is as the baby is developing in the womb, there's liquid in the womb and everything like that. Well, when it reaches a developmental stage, when all these alveolar buds and these bronchi start forming, the alveoli need to expand to start getting the process going to be able to take in oxygen and begin the respiration phase of development or set up for when it's birthed. I don't know. I don't think a baby can breathe inside the womb. I'm sure there's someone way smarter than me that has the correct answer to that, but I do not have the answer. But basically, what we need to know is in the beginning of gestation, um, as the baby starts to progress through gestation, um, phosphatidylcholine is going to come in and break up the surface tension around of the water or liquid surrounding the alveoli, and as a result, this allows the alveoli to expand and progress down the developmental stages. However, if phosphatidylcholine does not come in and break up the surface tension of the liquid surrounding, then that will lead to respiratory distress syndrome. And you see it in premature births quite a bit when phosphatidylcholine does not get a chance to go in and break up the surface tension to allow the alveoli to expand. However, if you guys are just trying to remember flashwords here, just remember respiratory distress syndrome and phosphatidylcholine. Um, I'm missing a big piece here. Sphingomyelin is replaced by phosphatidylcholine as gestation goes on. I completely just skipped over that little tidbit of information right there. But as I just said, phosphatidylcholine replaces sphingomyelin as gestation occurs, allows for the surface tension of liquids around the alveoli to break up, and as a result, the alveoli are able to expand and further develop the lungs. However, if that does not happen, then it will lead to respiratory distress syndrome. And that about covers it for now. I'm hoping we will be able to get to carbohydrates this week. And if everything gets super busy and we are unable to get to it, then I guess you guys are going to have to tune in for another video. So, uh, I don't know if you guys watched the whole thing, jumped around. Uh, I hope you guys got a little bit out of this. And again, as I said, if there are any questions, please reach out to me. And I am thinking about putting a list of practice questions together and possibly making a video on that, just so I can walk through a couple problems that you guys might see. And it also gives you guys a reference point to go back and hear me walk through some of these examples in case you forget how some of these problems go that we cover in tutoring. But I hope you guys have a good rest of your day. Study hard. I know we're on the brink of midterms here, so I wish you all the best of luck. Thank you.